This conference will now be recorded.
हेलो बच्चों, कैसे हो? All well. So this is in continuation with the biomolecules, whatever we were discussing in the last class. Let's have a quick review. Proteins. We discussed amino acid, the L family. All biologically active amino acid belong to the L family. Few amino acid essentials and non-essentials. I expect that you people have already made notes for the amino acid structure. It is definitely going to help. Don't try to memorize more than 5 or 4 amino acid per day. पहले essential करो, फिर non-essential करो. Amino acid are classified on the basis of character, essential, non-essential, acidic, basic, or neutral. You remember isoelectric point, it is the pH at which the particular amino acid neither moves towards cathode or anode, right? The isoelectric point represented as P chota I is going to be sum of K1 and K2. That is whenever you are going to add acid and base in a Zwitter ion. We did few examples based upon that also. Further, amino acid can join with COH from one side and NH2 on another side, and we can get thousands of amino acid, hundreds of amino acid joining to form di-tri-polypeptide. A polypeptide with no hard and fast 100 amino acid is going to be known as protein. Protein structure can be studied at 4 levels: the primary, secondary, and the tertiary and the quaternary, right? So that is going to be the structure of protein. You also discussed denaturation of protein, which happens with the addition of acid, base, matlab some chemicals, even you can add NaCl also, or even with the change in temperature, like boiling of egg. Properties of proteins, certain properties of enzymes, right, that you have already discussed in chemical kinetics. So let's move ahead today with the nucleic acid. This is another small but important topic. Small but important, you should not leave it. The nucleic acid is going to be broadly of two types, the RNA and DNA. We have to go with its structure under any circumstance, and please don't leave it right with it. एक second मुझे देना, just few seconds. So just few seconds. I'm just looking for some information if I can get it. Yes, beta, we have. Now it's okay. The living cell contains nucleoproteins, which contain protein combined with another biopolymer, nucleic acid, called polynucleotide. These are broadly of two types: deoxyribonucleic acid, DNA, ribonucleic acid. DNA is present in nucleus, mitochondria, etc. The RNA is present in nucleus as well as cytoplasm. The DNA is the major source of genetic information. How you resemble with your parents, your ancestors, that is going to be the DNA responsible for that, which is copied into RNA, the process known as transcription. As a chemist, I will not go beyond this in whatever you have done in the biology. The sequence of nucleotide contain the code for a specific amino acid synthesis. एक के बाद कौन से amino acid लगेंगे, that will be given by these nucleotides. The proteins are then synthesized in the process called translation. This transcriptional translation you people will be doing in biology. As a chemist, I do not need it. I focus here. We will be basically giving you a structure. Then the biologist will be using it. Nucleoside are the molecules important in which one of the bases, अभी बताता हूँ, purines or pyrimidines, all important, अभी आते चले जाएंगे, is bonded with a sugar molecule, the ribose and the deoxyribose sugar. A nucleotide is basically nucleoside तो है ही है, which has base and sugar. One phosphoric acid का derivative will come and that is going to be nucleotide. The nucleoside plus phosphate is going to be known as nucleotide. हम अभी इसका structure भी देखने वाले हैं. Please concentrate here. Nucleoside और nucleotide में क्या difference है? It is going to consist of a side, base and a sugar. Base and sugar. The sugar may be ribose, RNA में, deoxyribose, DNA. The second tense is nucleotide. Base and sugar is already there. What is going to be the additional thing? The phosphate is going to be the additional thing that is going to be known as nucleotide. पहले nucleoside बनाएंगे, फिर nucleotide बनाएंगे, then we will be telling you the polynucleotide. So let us discuss the masala, the constituent of nucleoside. पहले उनका base पढ़ते हैं, then how they are going to combine, that we are going to see. Bases, right? The first thing. The bases broadly are of two types, derivative of pyrimidine and derivative of purine. Pyrimidine further are going to be three types, uracil, cytosine and thiamine. Right? मेरे हिसाब से इनका स्ट्रक्चर नहीं याद करना है, but अगर आपकी मेमोरी है and you have very good rote learning, then you can memorize their structure. Pyrimidine are three types, the cytosine, the thiamine and the uracil. The purine is going to be of two types, the adenine and guanine, right? But whatever I'm discussing is important except the structures. तो अगर आपको पसंद आए, मैंने ऐसे याद किया, right? अगर आपको अच्छा लगे, it's entirely your choice. Purine is छोटा, pyrimidine is बड़ा. So purine are of two types further, adenine and guanine, and pyrimidine are of three named, cytosine, CTU, thiamine and uracil. Those who live in Chandigarh can easily memorize in this Chandigarh transport undertaking, but I'm telling you this will be important, CTU and AG, right? Now the DNA, हम DNA and RNA का structure कर रहे हैं. The DNA contains four bases. DNA contains four bases. The adenine, right? Thiamine, cytosine, guanine. And RNA में the right first three bases are same, but fourth one is uracil. RNA में thiamine नहीं होता है. So this is AUCG. Yes, you read right. मैंने आपको ऐसा बोला, ये वर्ड याद रखो. It will be helpful आगे DNA की सीक्वेंस बनाने में. At CG, DNA and RNA में the thiamine is not there. Thiamine की जगह पे क्या है? Uracil. You will be asked, just point out, list out the points of distinctions between the DNA and RNA के basis में. At CG ही याद रखना. This is definitely going to be important. अभी बताता हूँ. Adenine will be pairing with thiamine and cytosine will be pairing with guanine. ऐसे ही होता है. Nobody can change it. These bases अभी बताते हैं, इनके पीछे कुछ चीज लगेगी, ऐसे लगेगा, है ना? So these are going to be joined in DNA, right? Thiamine will be pairing with adenine with two hydrogen bond. This is the hydrogen bond. That is, I wrote two hyphens between A and T. ये DNA में होगा, ऐसा ही होगा. Don't change it. And cytosine always pair with guanine and vice versa. अगर एक DNA का C होगा, इसका G होगा, अगर इसका G होगा, इसका C होगा. And they are going to have it 3 hydrogen bonds. Biology वाले already know that how much important these are. So जो non biologist हैं, non med वाले, mathematics वाले, उनके लिए you have to follow. Right? What are we discussing? Structure of the nucleoside. फिर phosphate डालेंगे तो nucleotide बनेगा. The second thing is the sugar dirivose. मसाले डिस्कस कर रहे हैं ना, अभी इसको ज्वाइन करेंगे. This is the ribose sugar. Ribose pentagon, right? ऐसा ऐसा जानबूझ के इसको ऐसा बनाइए, ऐसा ऐसा इस प्रकार से. So here is oxygen, here we have OH, here we have OH, OH, and here we have CH2OH. जो मैंने नहीं बनाए वहां पे हाइड्रोजन लगा दीजिए. Beta D glucose, ribose. Now this is going to be in the RNA, sugar, ribose होगी. In DNA में it is going to be the only difference, oxygen नहीं होती है. That is why you call it deoxyribose. In my opinion, you should know the structure, right? बाकी आपकी मर्जी है. You should know the structure because of the obvious reason. We do not know why it is happening, but they are throwing more questions on the rote learning type. Although it is bad, but it is. So you have no choice. But rather you are lucky that I'm warning you this around 2-3 months before your examination is going to happen. Right, Ji? So what did you say? Here you will be having hydrogen. This is the difference between deoxy and this. Now आपको भी कराया Ag and Ct. DNA has At and Cg, RNA has Au and Cg. So nucleoside, this is the sugar. Here at the first carbon, you labeled it first. This will be holding one of the bases. ये bases कौन से हो सकते हैं RNA में? The AUCG में से कोई भी हो सकता है. Understand? Remember, the point of attachment of base with the sugar is one carbon का OH. In DNA, the base is going to be different, and this can be बेटे at CG में से कोई भी एक हो सकता है. This will be one nucleoside. Do you understand nucleoside? The sugar plus base. Sugar plus base. I'm just building points one by one, बेटे. So you can go through that. RNA और DNA में क्या फर्क आ गया है? This is going to be this, and थोड़े समय में यहाँ पे क्या डालोगे? Base. और ये base कौन से होंगे? AUCG. और ये कौन से होंगे? At CG. And second difference is, notice second carbon पे oxygen will not be there. चल सकते हैं बच्चों? Now see what I'm going to tell you more. In nucleotide आ गया. Sugar rings are attached to nitrogen atom, right, of a base by a bond between C1 of sugar, right, and nitrogen atom of a base. दोबारा पढ़ो. In sugar nucleotide, sugar rings are attached to nitrogen atom by a bond between C1 and C2. This bond is called glycosidic bond. That is, इस वाले बॉन्ड को वो क्या बह रहा है? Glycosidic bond. This one, the sugar and the base. Continue with it. The phosphate group is bonded to hydroxyl group of a sugar. Where will it attach? One carbon, the 2 carbon, the 3 carbon, the 4 carbon, 5 carbon पे. अब जो 5 carbon पे you have the CH2OH. This is going to be the phosphoric acid. You remember phosphoric acid क्या था? The OH and OH. this is going to be removed, right? I repeat, C1 of sugar and base, C5 of sugar with, you can say, phosphate, because phosphoric acid biologically बहुत ज़्यादा अगर आप देखेंगे it's quite strongly acidic, so he's showing it in the dissociated form. So what is this, my dear friends? The nucleotide has been arrived. The nucleotide has sugar, base, and the phosphoric acid. Now we are going to join these nucleotide further. These nucleotide further. ध्यान से देखो, right? You should draw it. This is one nucleotide. मैंने यहाँ पे OH बना दिया है. The C1, the base, इसको हमने adenine दिखाया हुआ, right? This is going to be sugar. 5 prime, हमने prime कर दिया, right? Prime is to differentiate between the carbon of the sugar and the base. Sugar की carbon को हम prime लगा देते हैं. This is 1, this is 2, this is 3, this is 4, this is 5 prime. Now ध्यान से देखिए, this is 5 पे phosphoric acid लगा हुआ है. अब ये एक हमारे पास दूसरा न्यूक्लियोटाइड आ गया ऊपर वाला. This is another nucleotide. See, इसकी 5 कार्बन पे जो phosphoric acid लगा था, that is having OH. शुगर का 3 कार्बन भी तो फ्री है ना, शुगर का 3 कार्बन भी तो फ्री है, वहां पे OH है. फॉस्फोरिक एसिड का OH एंड शुगर का 3 कार्बन का OH is going to lose water molecule to form this linkage. This is 2 nucleotide, this plus this is going to be the dinucleotide. Try to understand what has happened. It's very easy. Now सिर्फ होगा क्या, अगर RNA है, right? A, अगली बारी U आ सकता है, C आ सकता है, G आ सकता है. That you or me do not decide, the mother nature decide. And RNA में, here we will be having OH, right? And DNA में these bases can be A, T, CG हो सकता है, and there we will be having this. So my dear friend, if I ask you in another way, this is nucleotide of RNA or DNA, your answer is this is the dinucleotide of the DNA, because यहाँ पे oxygen is not there. Do you understand once more what is I have drawn? Same thing. That means अगर हम easy language में आपको बताएं, you know, ये थोड़ा सा broaden कर दिया जाता है, मतलब you are watching it from this side. At this point, what do you have? Base. I'm just writing a box. It may be having OH or H. Base can be ATCG or base can be AUCG, RNA and DNA. This is going to be the phosphate, right? Here, I'm just symbolically representing the phosphate unit will be formed. And अगर ये join किया तो ये कौन सी कार्बन होगी? 3 प्राइम. ये कौन सी होगी? 5 प्राइम. ये कौन सी होगी बेटे? The 1 प्राइम that is going to be formed. Do you understand? Watch it another way. It's the same thing. We are just repeating. Nucleotides are joined by phosphodiester linkage. This is the phosphodiester linkage, my dear friends. Phosphodiester linkage. ये phosphodiester linkage है. You are able to see that, right? मैं just जानबूझ के कलर कॉन्ट्रास्ट बना के आपको प्रैक्टिस के लिए बताने वाला. The phosphodiester, removing the color, right? So that is going to be happening here. आगे जा सकते हैं. Please, 3 prime, 5 prime, 1 prime. 5 prime of 1 and 3' of another पेंटोज शुगर में बॉन्ड बनेगा. कभी कभी इजी वे में राइट करने के लिए we also write in this way. The nucleic acid is abbreviated by one letter code with 5 end of a chain written on the left. Right? 5 end of the chain written on the left. For example, a tetranucleotide having cytosine, guanine, thymine, cytosine, guanine, thymine, adenine. It is going to be represented as, देखो पहले का the 5' will be free ना, इसका 5' will be free, that is being written here. Now पहले का the 3 will be holding दूसरे के 5 को, so इसका 3 is holding इसका 5 with phosphatized 3 5 3 5 3 5 and so on will be happening. पहले का 5' and last वाले का the 3' is going to be available for bonding. Did you notice? इसका 5, इसका 3. That is the code of the nucleotide is written. You know, this is the fundamental of life. This happens. Tomorrow, अगर आप RNA का को synthesize कर रहे हो, virus को देख रहे हो, bacteria को देख रहे हो, sequence will be this, but the sequence will be changing. Basis की. Test for protein, not for your CBSC, but you should see to that. Sample to be tested, for example, let it be egg white. Add little bit of NaOH, shake thoroughly. Add 1% of copper sulfate by drop. If you are going to have violet color, that is going to indicate presence of what, my dear friends? The presence of proteins or amino acid हमारे पास आ जाएंगे. There we are done with structure of the nucleic acid. Now RNA तो single strand ही होगा, DNA के structure में there is something special going to happen, right? The DNA structure is a duplex structure consisting of 2 strands of polynucleotide chain coiled around each other in the form of a double helix, like 2 snakes, right, coiling around each other. That is going to be the DNA का structure होगा. You can see with various figures. This is going to be the DNA and this is going to be the RNA. अभी पहुंचते हैं यहाँ पे. I hope first thing, DNA has a duplex structure consisting of 2 strands. The 2 strands are marked as 5', 3'. आपको याद है ना 5', 3'. शुगर की कार्बन को डिफरेंशिएट करने के लिए हमने प्राइम यूज किया, which indicate the free hydroxyl group of terminal deoxyribose unit are present at 5 and 3 position respectively, that is anti-parallel. एक का 5' और एक का बेटे 3' ऐसा पाया गया. That is a linear polymer has a 5' hydroxyl group at one end and a free 3' at another end is going to be there. Non-medical वालों के लिए more important, biology वाले already know that. So I'm repeatedly saying this word again and again, my dear friends. The nucleotide of each strand are connected by phosphate ester bond. This forms backbone of each strand, right? Phosphorus, sugar, base, right? Sugar, base. Phosphorus, right? Sugar, base. Phosphorus, sugar, base. He's saying this thing is going to be repeated. I hope you understand. These bases might be different. The bases ATCG points towards each other while sugar and phosphate form the structural frame of outside of the duplex. Abhi karwa do. The bases are thus the steps of a spiral case and sugar phosphate is the railing. देखो ना, this is sugar, phosphorus, the base, the phosphorus, the sugar, बाद में fill कर देते हैं, the phosphorus, the sugar and this, right? Let us little bit change it, right? जब काम करना ही है तो अब इसी को मॉडिफाई करेंगे। अब आपके मन में जो भी आ रहा है, यू कैन स्पीक लाइक दिस, A, P, T, G, C. You could have said anything. Now what happened? सामने तरफ भी शुगर फॉस्फेट, यहाँ पे शुगर लिखना था बेटे, एक सेकेंड है ना। So this is going to be the sugar आना था, right? Now it's correct. Now what will happen here is सामने तरफ से we are going to have, right? एक सेकेंड, A है तो यहाँ पे T आएगा, यहाँ पे T है तो इसका A आएगा, इसका T है तो इसका A आएगा, इसका G है तो इसका C आएगा। They will hold each other by hydrogen bonding. The bases are the steps of a staircase. आप एक सीढ़ी चढ़ रहे हैं, and sugar phosphate is really now, which is straight, twist it like this. That is going to be known as double helix structure. Watch again, the double helix structure, right? The order in which base occur is called the base sequence. This is fixed in every organism. ये हमेशा डबल बॉन्ड और ये ट्रिपल बॉन्ड हाइड्रोजन बॉन्ड से होते हैं। No other combination is possible strictly. If you want to create any other combination, please go to some another heavenly body. Right now, we do not have any information. Who knows, tomorrow you will be going to another heavenly body and you will be discovering new type of life, new type of basis. But right now, no, we stop here. The two strands are called complementary because the base sequence of one always determine the sequence of another. See, he can tell you sequence of one, ऊपर वाला, and he can ask you दूसरा, बस ये याद रखना, at CG, at CG, CG, GC, at TA, at. This will be the sequence which will be happening here, right? This is the way it will be following, it will be happening. So you can see anywhere GC, AT, 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 the DNA 5 prime, 3 prime, 5 prime, 3 prime. Understand? Notice DNA double helix, RNA single helix, at CG, AUCG, the deoxyribose and ribose. That will be the difference. Important. It is deoxyribose sugar, sugar is ribose. You should know the structure, we have drawn it. It is double helix, it is single helix. Base pairing, no base pairing. And this is responsible for the transference of genetic material. This is responsible for the protein formation. You can add more words here with you. Right? One more picture of this. See the beauty of the life. You, me, any living organisms has this type of DNA. Hats off to the engineers who gave us techniques to visualize it. You know, without physics, you will not be able to see the structure of this. That is molecular structure के लिए electron microscopy जरूरी है. The advancement is being done, अभी और होगी, but एक जगह से आगे हम नहीं जा पाएंगे. So we started here, we are back there. There's a structure of what the nucleotide. This is OH or the H, OH in RNA, RNA and H in the DNA. The basis will be different. Note, DNA helices are normally the right-handed. Right-handed helix, जो हमने पीछे भी बात की थी. On heating, the two strands separate out because hydrogen bond will break. That is known as melting. And on cooling, these mix again. That is annealing. The temperature at which two separate is called melting temperature, which is specific for each specific sequence. Why? Because number of double bond or triple bond will be different, and hence this is going to be different. You can say this is a signature of DNA of different beings. If melting point is higher, then Cg contents are more at its 3 hydrogen bond, resulting into what? The stronger hydrogen bonding is going to be there. Right? We did this also. RNA has similar, except it is the single strand पाया गया है. Right, बेटे? थोड़ा सा, but not less important. I will say this topic is more important for your general knowledge, for your day-to-day life, rather than examination point of view, sir. Because tomorrow you study science or you do not study science, you are studying whatever, you may be doing whatever in your life after 20 years, 50 years, but these words are going to be important. It has been observed that certain compounds are required in small quantity in our diet, but their deficiency may cause specific disease. These compounds are called vitamin. आपकी जनरल नॉलेज के लिए इंपॉर्टेंट, स्कूल के लिए तो है ही है. Most of the vitamins cannot be synthesized in our body, but plant can synthesize almost all of them. These are considered as essential food factor. However, the bacteria of the gut can produce some of the vitamin required by us. For example, vitamin B complex. So whenever you take antibiotic, that is going to destroy the flora and fauna of your gut intestine. So you lose that friendly bacteria also. So the problem is definitely there. So your doctor will be giving you some pro and prebiotic to restore that. All the vitamins are generally available in our diet. Generally kaha 100% nahi bola. Different vitamins belong to various chemical classes, and it's difficult to define them on the basis of structure. These are vitamins regarded as organic compounds required in diet in small amounts to perform specific biological function for the normal maintenance of optimum growth and the health of the organism. If you do not have particular enzyme, your particular process will not happen. You will be suffering from certain disease. Vitamins are designated by alphabet A, B, C, D. Some of these are further subdivided as subgroups B1, B2, B12, etc. Vitamin D3. Excess of vitamins is also harmful. And vitamin pills should not be taken without the advice of a doctor. Never ever forget this thing in your life. Right? I remember a case. So I was sitting with a doctor. So he was just reprimanding some lady. So somebody told that lady that you should be taking vitamin D3. And she took vitamin D3. डॉक्टर ने कहा था, but वो डॉक्टर के पास दोबारा गई नहीं. And that lady was taking vitamin D3 tablet for 3 or 4 months daily. डॉक्टर ने बहुत बुरे तरीके से डांटा. Do you know what you are doing? So do you know कि विटामिन D3 की 3 महीने आपने रोज डोज ली है, आपने क्यों किया है? द ओरिजिनल डॉक्टर जिन्होंने एडवाइस किया था उन्होंने तो सही एडवाइस किया होगा, इन्होंने सुना नहीं होगा, उन्होंने कहा होगा मे बी वीकली लेना है for 1 month, after that it should be stopped, it should not be taken more than that, but again you should be listening to the advice of a doctor. That lady took 3 years vitamin D3. Ask from your doctor 3 महीने continuously 60,000 international unit अगर कोई vitamin D3 खाएगा तो उसका हैप्पी बर्थडे होने के कितने चांसेस हैं? Anyway, the term vitamin was coined from the word vital plus amine, since the earlier identified compound had amino group. Later work showed that most of them do not contain amino, so the letter E was dropped and nowadays vitamin is used. पहले amine था, अब E उड़ा दिया, बस उसको differentiate करने के लिए. Vitamins are classified into two groups depending upon their solubility in water or fat. fat soluble, which are soluble in fat and oils, but insoluble in water are kept in this group. These are A, D, E, K. They are stored in liver and fat. That is, fat जहां पे होगा वहां पे ये पाए जाते हैं. Water soluble, B group and C group are soluble in water, so they are grouped together. Remember, water soluble vitamins must be taken regularly in diet. Diet कहला दवाइयां नहीं कह रहा, because they are excreted in the urine and cannot be stored except vitamin B12 and water. You just remember this thing. Water soluble याद कर लो. B and C. Rest all are the fat soluble. B and C. आपने सुना होगा जो सेलर्स होते हैं उनको विटामिन सी डेफिशिएंसी हो जाती है बिकॉज उनकी डाइट में विटामिन सी आता नहीं है एंड दैट इज वाटर सॉल्युबल, तो वो यूरिन के रास्ते से बाहर चले जाता है एंड दे सफर स्कर्वी एंड अनदर डिजीजेस. Very important, as I said, from school point of view you leave it, but from your general knowledge point of view you should do that. विटामिन ए किस में पाया जाता है, डेफिशिएंसी क्या करती है? Vitamin B1, B2, B6. You will read it at chill, easy. Vitamin B2, riboflavin, right? It is chiliasis, fizzling at corner of mouth and lips. अक्सर हो जाता है, right? आपके मुंह में boils या छाले जिसको हिंदी में बोलते हैं पड़ जाते हैं. That is vitamin B2 की वजह से होता है. That is riboflavin. Vitamin B12, pernicious anemia, vitamin C, scurvy, bleeding gums, and it is also found to be responsible for building up your immunity. Vitamin D, very important. Nowadays, because of the current lifestyle, we people do not go to parks and in sun, right? Our skin is not exposed to sun, which synthesizes vitamin D3. So everybody, you can say most of us are deficient in vitamin D3. Probably a study is saying that the current coronavirus, right? The persons who are having healthy vitamin D3 in their body, they are suffering less, and those who are not having vitamin D3 in their body, they are suffering more. Probably that could be the reason, anyway, you people will search for it, that Europe and America suffered more because of the corona. Probabilities. Vitamin E, right? Muscular weakness. It is responsible for cramps. Vitamin E. Vitamin K. It is used for increasing blood clotting time, my dear friends. Right? So that was an important, easy chapter known as biomolecules. Easy chapter. So your job is to go through that, right? Read your NCRT, make your NCRT a base easy, but it is definitely a scoring chapter, my dear friends. So 70 out of 70 will be given by this. JEE mein, right? In particular JEE mein. So it might give you those 5 or 10 marks which can pull your rank almost by 1 lakh. I'm not joking. So mark my words seriously. Structure देखना amino acid के, right? Who knows, someone from you is creating these nano robots. We need you people, right? We stop here, my dear friends, and see you again, bachcho, with next topic. Good luck with it. Bye. Thank you. Signing off. Good luck.